Welcome to Examining Evolution, everyone. Thanks for being here. I'm here with Sal Cordova, who's done a lot of work on looking in, researching evolution. And tonight we're kind of going to talk broadly about what is wrong with evolutionary biology. And Sal's going to share some of the things that are on his mind about this. So go for it, Sal. Welcome.
Well, welcome and thank you for having me, and greetings to everyone in the audience. Tonight is going to be kind of a practice session because Rebekah and I have never done a show quite like this. I mean, we've had some other shows, but this one is being hosted on her Examining Evolution channel. Our last talk was on Cures for Doubting Thomas on her Bread of Life channel. We did have like a couple sessions talking about her visits to some of the internet sites. And I said I wanted to try to do something a little more methodical and explore ways to communicate some of the things that have made me skeptical and eventually really rejecting evolution. The more that I studied it, the more that it seemed like just a horrible theory. I mean, compared to others, I mean, it may look beautiful to some. I was raised in the Catholic Church, and I went to public school, and I accepted evolution, and I accepted a version that looked very optimistic. I kind of liked the idea that simple life forms could evolve into something better. And the expectation was that we would just keep evolving and getting better, and the whole universe would get better. So that's kind of the naive view of a child. And I thought, well, that's a really cool idea. And I'd see these pictures of the progression from microbes to becoming like a fish, and then it becomes like a reptile, and then a mammal, and the mammal becomes an ape-like creature, well, apes, and then or primates or whatever, and then you see it become a man. And I said, that's really beautiful. And I said, if that's how God works, I think that's just a great way that God could work. So that was where I was. And after I became a Christian, I still had accepted evolution, and then I didn't quite understand how it could be reconciled with the Genesis account. And I said, well, okay, well, sometimes we harbor ideas that aren't really compatible, and yet we can accept both of them. And that's sort of where I was as a young Christian and a sophomore in high school. I mean, you just sort of accept whatever's thrown at you, and sometimes you don't really think about it. I didn't. It was only slowly I realized these aren't compatible ideas. And I ran into a tract that talked about the complexity. And I said, yeah, it doesn't, I don't think evolution's feasible. Now, actually, there was something metaphysical that actually persuaded me. I said that it's hard to imagine. I said, at what point can chemicals just keep interacting and then it becomes self-aware? It starts to feel like it has a soul. I said, the chemicals that make me up now are not exactly the same chemicals that made me up when I was a child. I mean, it's a different set of atoms. I mean, we know the cellular processes. There's lots of turnover of the chemicals and molecules. So you take nutrients in, and then it gets cycled out of you and replaced. I mean, there's some parts of the brain that they believe are relatively persistent in terms of the same atoms. But I said, something more than just the mere molecules seems to make me up. But then years later, after I'd gotten 3 science degrees, I began to have a crisis of faith, and I covered that in our last show. And one way that the Lord answered my prayer when I was looking for evidence of God was to study intelligent design. So it began with studying the problem of abiogenesis. Now, evolutionists will say abiogenesis is not the same as evolution. So I'll say, okay, I'll accept your definitions. That's fine. But it became evident that a miracle had to create the first life. And in Darwin's Origin of Species, one of the versions, he postulated a creator that created the first life. You'll see that toward the last chapters. Now, historians will say, well, did Darwin just insert that because he wanted to appear more like a theist, even though he was an agnostic? Or did he do that to kind of appease others, especially his wife, who was a professing Christian? We don't know. But it is an Origin of Species where he did suggest that the first life or first few lives were created. And you can Google that, you'll see it. And it's really kind of amusing that he says that, because technically then he would be some sort of creationist by today's standards, if we take that literally. Now, so then the theory of evolution is what happens after that through a process of common descent. So I was I'm fine with people. I actually encourage creationists to make the distinction between abiogenesis and evolutionary theory. But yes.
Can I pause you there? And yeah, I run into that all the time, and I'm fine with making the distinction in order to appease them. But really and truly, the problems it's the same type of problem for abiogenesis and evolution, right? Even if you get abiogenesis, you still have the same types of problems when it comes to evolutionary development. Would you agree with that?
Not quite. Not
Okay.
quite. Because now you have something that's alive and reproducing. Most people have said that barrier to get the first life is just enormous. Lynn Margulies said that she thinks it's a bigger barrier to go from non-life to life than to evolve, like, say, a single cell into a human being, because the leap is that gargantuan. So that there's no things like inheritance in abiogenesis. So it really is, there's justification to call it different phases of a problem of what I call a naturalistic explanation. So the naturalistic explanation will have to be abiogenesis plus evolution. But the point was, and Jerry Bergman did a good job of this, he said, okay, if you have to accept, as Darwin, if you take what he said literally, and again, the caveat is he was a known liar, and he may have just said that to say it, but let's just take what he said literally: the creator created the first life form. The issue is then, if you admit one miracle, why can't you admit more? If you're willing to admit a miracle of the first life, well, then where does it stop? Because if we can demonstrate, as I will try to tonight, that there are miraculous steps, like, say, going from prokaryote to eukaryote, and it's like, okay, so here's another miracle that evolution needs to make it work. And so evolutionary biology then is in a kind of the unfortunate situation that they're trying to give a naturalistic explanation for many of the features of life. But when it needs miracles to make common ancestry feasible, how is that fundamentally different than creationism, except maybe the timeline and chronology? Because they may say they don't need miracles, but when I confront them on some of the things that we'll talk about tonight, they're like, I don't know how that happened. I'm just like, okay, that's fair enough. We don't know a lot of things. But if you're asserting this is true, and you have evidence that you have no way of ever explaining this in terms of natural processes, what I mean, I could show you all the accepted laws of physics and chemistry. I could show you principles of cellular biology. If you can't explain it in terms of that, then your belief in evolution is faith-based. It is not based on fact. And Rob Stadler, and Rebekah, you've read The Stairway of Life by Change Tan and Rob Stadler. There it is. And by the way, she must have read this cover to cover, because she read it all the way to the acknowledgement section and found my name there. I was very flattered. So thank you.
Yeah.
Rob Stadler wrote another book about good and bad science, or really high quality science and low quality science. And evolutionary biology is rated as low quality science. That means it's filled with lots of unverifiable assertions and claims. You compare that to, say, oh, are you looking it up?
Yeah, I've read that book too, and I love that book. It is
What was the name of it?
It is the scientific approach to evolution.
Wow.
Yeah. I recommend, I actually made a video about that book, Sal. I think that's a book every student should read.
Yes. Did it mention, did most of, I mean, I would presume it didn't talk about God very much. It was just straight
No,
science.
not at all. In fact,
Yeah.
when I ordered that book, I was actually just looking for books on evolution, not books, that had, a creationist or, not just books on evolution. And I found that one. And, I started reading it and I was like, wow, this is a really great book. I'm not used to people being so fair about evolution.
Yeah.
The assumptions were not built in, but so he is very fair. He does present good evidence for evolution. And like you said, the book really shows like what criteria scientists use to establish, like, should we have confidence in this? Is it high confidence science, or is it something that's low confidence? And so, like you said, he said there's 6 things that he lays out.
And this actually will start to have impact on our medical science. For a long time it didn't, but when they started using evolutionary theory to talk about what parts of the genome are functional or not, I said, you guys, you have no business getting in on this discussion at all. It is up to those who are studying the cells and determining experimentally what actually works or doesn't. On what basis, except evolutionary theory itself, are you saying, like, the ENCODE project at the National Institutes of Health, which, by the way, was like a $500 million research project, if you include the follow-ons, like what they call the roadmap epigenomics. I said, on what basis are you saying this is junk? They'll say evolutionary theory, and they'll claim this is the best theory. I said, well, maybe the theory is problematic if we're finding that it has a lot more function than you claim. I said, you have no business taking a flimsy theory, a theory that has weak justification in terms of physics, chemistry, cell biology, etc., and then saying that, oh, this section of the DNA can't be useful for anything. When I was at the ENCODE conference in 2015, I was wondering what all this was about, because the evolutionists were really upset with them. One of them, Dan Grauer, is calling them crooks and ignoramuses. I'm just like, what's that about? I mean, those are the sort of things that they would say of creationists, but they're saying this of fellow scientists who are cell biologists, biochemists, serious cancer researchers. And so I went to this conference. I for those of you who don't know who I am, I was a research assistant for Dr. John Sanford for 7 years. He's a famous geneticist who was an atheist. He became a Christian and then a creationist. And he was very interested in the design of the cell and this, his theory of genetic entropy, we can also call genetic deterioration. And he said, Sal, go to the ENCODE conference. And I went. It was a 3-day conference, and I said, what's all this? Why are they so interested in what the evolutionary biologists call junk DNA? And the researchers would be coming up one after the other and saying, okay, I'm working on this particular disease, and it seems to be caused by mutational damage to areas that we call, that the evolutionary biologists would call junk. And there were studies that showed that 90% of the heritable diseases or diseases related to DNA were in the so-called junk regions. And so some quarters of evolutionary biologists were very upset hearing claims and then also supporting evidence that the human genome could be very functional in ways that we don't understand. There's been a follow-on project called 4D Nucleome. And I've gone on record and said, I have my own biases. I tend to think it probably is functional. We don't know. It could be that it had original function that we've lost since the fall of Adam and Eve out of the Garden of Eden. We just don't know. But it's not for evolutionary biologists to tell a cellular biologist that this can't work because evolutionary theory says it shouldn't. It's the job of scientists to experiment and determine and observe. And the ENCODE consortium, and now the 4D Nucleome, and then we have so many follow-on projects, I can't even name them all anymore because there's so many. They're the people that should be doing the high-quality science to determine what is useful and is not. And so this is an example where low-quality science of evolutionary biology is actually probably hindering the advancement of finding cures. And I just want to tell them, you guys need to butt out. You're not helping. And you're also, it's not helping that we're driving creationists out of the biology curriculums when we need more researchers. They should be welcomed. And yet they're being tarred and they're being persecuted because they think maybe God created biology. I mean, why can't, at my graduation from Johns Hopkins, Dr. Ben Carson was supposed to speak at my graduation. And he, because he was a Christian, there was an outcry and he couldn't speak. But he's a medical doctor and pioneered. I'd classify him as a scientist for what he did. This is not healthy for science, that we're using things like evolutionary biology to discourage people that reject evolutionary biology from the science curriculum. This is not healthy. And then the other thing that is not very, it's a little sobering. It may be that the human genome is slowly deteriorating in terms of function. I've surveyed a lot of the literature by evolutionary biologists and geneticists that are studying human heritable diseases, and all of them uniformly, to the best of my knowledge anyway, are saying that our genomes are getting worse with each generation. You see, this is not consistent with the idea that we evolved from
progressing.
Yes.
Right.
Because the evolutionary biologists will say, well, no, it's just radiation. It's not that we're really necessarily increasing in complexity. But I have experimental evidence that if we evolved, which I don't believe we did, that our proteins are much more sophisticated than those, like, say, in a yeast. We see this in something called post-translational modifications, and someday we can get into that. But we are very special creatures in terms of the way our proteins are built. We are special. And I mean, if we just wanted to just look at the biology and say, are we special? And it's like, yeah, Yeah, we are.
Yeah.
From the, people don't want to, I don't know, because it's really funny that some, there's been a tendency in naturalists to try to say we're not special, because if we are special, that suggests something miraculous. And it also suggests we are special and therefore special in the eyes of the creator. And they don't want to, any hint of that is like, no, this is makes us feel uncomfortable, because if we are special in the eyes of the creator, we're accountable. So let me see what Dessel Dre says. If Sal
If
saw evidence of
self,
men that appear to viral and show signs of degradation, have pathogenic function, and they're switched on, would he accept them as junk? Yeah. So there's stuff that's pathogenic, but we can't just uniformly say 90% is junk. So do you, one example was the Alu element, Alu repeats. They thought that was just junk insertions, because we do have junk insertions of Alu repeats, but because we have some junk doesn't mean it's all junk. And I wrote a chapter in the book. I could try to contribute a chapter to the book Contested Bones. It got chopped down to 2 pages. And I was like, Dr. Sanford, I spent a month on this looking at the Alu repeats. And it turns out those are very sophisticated memory devices. They're not junk repeats. So thank you for the question. So there's
Dessel has like 3 other questions here. So I don't know if you want to take them now or you want me to save them.
Let's save it for later.
Okay.
Because
you don't want to get too far off track, or
no.
Okay.
So that's where that's a long introduction, and I'm sorry for the monologue here. I have some slides and I have some things to point out, but can you show that paper of by John Welch, What's Wrong with Evolutionary Biology?
Yes. I don't have it pulled up electronically, but I just, yeah.
So I recently co-authored a paper on fitness maximization, dynamical systems and fitness maximization in evolutionary biology. And the conclusion of the paper was that one of the central concepts of evolutionary biology was not robust. So we tried to use polite language. We tried to use something not too provocative. And we succeeded in not being too provocative, because we quoted papers like that one written by an evolutionary biologist. So that the title of that paper that Rebekah held up is What's Wrong with Evolutionary Biology? That wasn't written by a creationist. That was written by an evolutionary biologist criticizing his own discipline. And there turns out to be a good number of papers like that if you're willing to comb through the library. Popular science writers and promoters don't really like titles like that, because they want to say, oh, this great discovery. They're not eager to say, oh, we have a great discovery. Evolutionary biology has stuff wrong with it. And so when I was reading this paper, I was just laughing. I was reading it with a biology friend named Emery Moyna, and he's a creationist biology student. He has to go by a pen name because to try to protect himself so he doesn't get kicked out of graduate school. I'm expecting he's going to graduate at the top of his class, by the way, because the saying is among us creationists who are graduate students is you got to be better than your peers because you are a creationist. You have to fight a little harder, because if they've ever found out you're a creationist, it's like, well, gee, he was a really good student. He deserves to graduate even though he's a creationist. I mean, that's and God bless Emery, that's what he's doing. He is excelling and he is trying very hard to be an excellent student. That's what I encourage all creationists. And I know it has to be tough because he has to take classes in evolutionary biology. I had to take classes in evolutionary biology. So where was I headed with that? Anyway, we were reading that paper and we were laughing because like every other paragraph or so, he'd highlight a problem the creationists have highlighted. We would just laugh. It's like, oh, gee, oh, you finally realized that that's a problem. It's nice of you to acknowledge it. And there, that paper tried to offer some solutions to why there are all these complaints and maybe how to stop them. And I said, well, maybe, rhetorically it came, Emery and I said, well, maybe the problem is the theory's wrong. Maybe that's why there's so many complaints to revolutionize the field and overhaul it. There's now this called the third way of evolution, this trying to find other means of mechanisms of creating complexity other than so-called natural selection. And really that's a misnomer because what Darwin conceived is neither natural and it's not really selection. It's maybe a better term is fantasized selection because what happens actually in terms of experiment and observation is not consistent with the way he conceptualized things because we're finding what happens naturally is loss of complexity. So his idea of constructing things by means of increments, that doesn't really work well. And I'll give an example of that. So Rebekah, talk to me.
Yeah, well, I think, I would love to hear some of the things you shared. You said that it takes miracles, like there are miraculous steps needed. And I'm very interested in seeing some of the one of that video that you talked about.
Okay, so I published 3 papers on the topoisomerase 2 enzyme, 2-alpha enzyme, and I'm going to show a video that shows the 2 varieties of topoisomerase. How did I get published? Well, you join a lab that has someone that really knows what he's doing because I couldn't do it myself. I was an engineer in the aerospace and defense industry, and I found someone that had studied the topoisomerase enzyme for 17 years. 17 years on this one enzyme. And an enzyme is a protein, and proteins are made by genes. Now, we estimate they're anywhere between something like 20,000 genes, and this is only one of the proteins from one of those genes. And they have other things like, if we include the alternative splices, 500,000. So we're only looking at one alternative splice, what we call an isoform. I think that's the right term. Correct me if I'm wrong. It's been a while, I'm not that persnickety about terminology. This is only one, and a lot of people have been spending their lives, entire professional lives, on one protein or one enzyme. And this is a very well-studied enzyme. The reason it's very important is when we try to disrupt the way it works, its function, that's the basis of chemotherapy. So when we want to kill cancer cells, we throw chemotherapy at it, and it will disrupt the topoisomerase in the cancer cells. And unfortunately, it also disrupts the topoisomerase function in healthy cells, and that's why people on chemotherapy get very sick. Part of my work was trying to find ways we could better target the chemotherapy methods with the topoisomerase enzyme. And it was really interesting as we were doing this work. I was like, this is really good evidence of a creator. And so the first thing I'm going to show is what the topoisomerase actually does. And you could see that when it's disrupted, it's not going to, DNA will not function very well. And that's why the cell dies when you throw chemotherapy at it. So this is a video of the topoisomerase enzyme, and let me start sharing it. It's only 2 minutes long. And this will lead to one of the many, many problems with evolutionary biology, because I will talk about the evolution of topoisomerase going from prokaryote to eukaryote, and it leads to a lot of problems. And it's a monkey wrench. So when you ask me, Rebekah, what are the miracles needed? in common ancestry. This is one of them. So the first thing is, and this is very interesting, parts of it could be classified as abiogenesis, but then parts of it might not be. But before we make those distinctions, let's just see what the topoisomerase enzyme does. So let me share the screen here and get the Chrome tab going. All right, here we go. I'm going to mute my mic so we get better audio.
Let's consider what happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase I enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase II enzymes do the same thing but with both strands of the double helix. Topoisomerase II cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase II can remove the supercoiling, 2 twists at a time, to yield a relaxed circle.
Great video. It's amazing. It's hard to imagine how that could have developed by a natural process.
Sorry, I was muted. And one thing I like to say, it's not just that we can't imagine it. As a matter of principle, it shouldn't happen. It's, so now I'm going to give a pointer. Yes, it is hard to imagine, but it's hard to imagine because it's unrealistic for us to expect it would happen naturally. And what I mean by that is, we wouldn't expect a tornado passing through a junkyard to assemble a 747. All the pieces have to be in place. And let me give you some examples of that. Maybe we could show the video again and I'll pause it and we'll talk about some of the stuff. Because I think it'll be more powerful if we talk about it. So we could classify we could, I'll show how this can relate to evolutionary theory, but let me see if I can get this going again. Let me get my buttons all in order here. And thank you for your forbearance here, Rebekah.
And Spim says it's hard to imagine when you don't try it all.
No. This is,
if it's science, we shouldn't have to imagine. That's the thing. I shouldn't need my imagining.
Yeah, this is like imagining a tornado passing through a junkyard making a 747. As a matter of principle, it's just ridiculous. It's not a matter of ignorance that we're saying this is unlikely. It's what we know. We know things don't assemble like that, and I will demonstrate that as we actually go through the architecture of the topoisomerase enzyme and examine the amino acids and see all the sequence that you need to be able to create something this sophisticated. It's not a matter of our lack of imagination. It's the fact that we know that we can't just throw random sequences and make anything like this. And then someone will say, well, what about natural selection? I'll show you why natural selection won't work.
Happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands.
Okay, so this is really amazing. Just I, this is just a property of DNA. Everything's just so crowded there in the cell, and a lot of things can get tangled up, and so you have to untangle it. And so, in the process of either replicating or transcribing, stuff will get tangled. You have to untangle it. So that's what the topoisomerase does.
As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together.
So the importance of this is when you're not able to untangle the DNA, the cell dies. So that's why we had the chemotherapy. We're going to prevent the DNA in the cancer cells from being able to unwind, and therefore we kill the cancer cell.
This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase I enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase II enzymes do the same thing, but with both strands of the double helix. Topoisomerase.
Okay, let me stop here. So let's say you have an enzyme and it figures out how to cut the DNA, but doesn't know how to stitch it back together. That would be really bad. That would shred the genome. So how's natural selection going to deal with that? If you have something that's a cutter, but not something that reconnects, what we call ligate, let me just use the word reconnector. Let's say you don't reconnect and you just have a cutter. Well, that doesn't work. Cell's dead. Or let's say you have a reconnector, but you don't cut. Well, that's no good. Cell is dead. And let's say, go ahead, Rebekah. I could see.
I don't want to interrupt you, but I guess since I already have, I will go ahead and say this. There's nothing like who had the foresight to know, and I say who because I don't see how a natural process can have the foresight to recognize that this tool was needed to untangle, right? Like you've got to have some kind of agent that sees the need of this, right?
Exactly. And people say, what about natural selection? I said, it's kind of hard to select something that's dead. I mean, all right, you have the chemotherapy. Chemotherapy it disrupts this. So this is life critical. If there's no topoisomerase, most life forms that I know of will die. Now, someone might say, okay, we have a phage, but I'll say that's not cellular life. That's viral. Okay, so when I talk about life, I'm talking about cellular life. So if you have just the reconnector, but you don't have a cutter, that's bad. If you have a cutter, but no reconnector, that's bad. So you're not going to evolve this in pieces. The other thing is, how does it know where to untangle? And watch this move. Okay.
2 cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break.
Okay, so it has to have, I don't know what you call a repositioner, where it would, so it has the cutting step, the untangling step where it passes, one thread between the cut, as you saw there. Did you see that? And then the reconnection step. That's at least 3. We could also say, how does it know to do this when it's tangled? It needs to sense the tangle. So if it doesn't have all those 4 things, the thing's dead. So if they say natural selection can make this, I'm just like, how could it make this? So let me just stop here, and I'm going to maybe go to my slides. They'll say, well, that's not, evolution. I'm just like, okay. I wrote a paper that was criticizing some of what the evolutionary biologists were saying, where they said this evolved independently in several lineages. I'm just like, yeah, right. How did it not die in the process? You're just asserting that. So these things will pass through peer review. They'll be approved by evolutionary biologists, their editorial boards, and no one bothers to say, you didn't justify how this didn't die in the process of evolving. If it's independently, they could probably say, well, maybe that's an origin of life problem, but it's like, okay, yeah, I'll show you some other problems with that. So
why do you think they're not saying that? Why are these papers getting through peer review and no one's asking critical questions?
I think it's because they're just accepting it by faith. And they probably think, well, that's someone else's problem. He's going to figure it out. I think they have a large amount of faith that someone's going to figure this out. That it had to happen because there's not any miraculous intervention. So I want to give space to some of these theistic evolutionists like Joshua Swamidass. They'll believe, well, it's natural. I'll say, well, justify that in terms of all that we know in terms of cellular biology, biochemistry, organic chemistry, physics, you name it. And how about a dash of logic to boot? And they can't. But they just have so much faith that someone else will figure it out, and that that's the most that I could say. And there has to be something about like, I see this, especially for scientists that have done this all their lives. How would they feel about themselves if they've paid their mortgages and built their reputations on an idea that's fundamentally untrue? So, that's a lot to say, oh my goodness, all that I spent my life on was false. That's kind of hard to deal with. So, I think sometimes the creationists tend to say, oh, these people are just really immoral, whatever. I don't go there. I say it's probably even more basic. It's like, well, this is something that they've invested their lives in. It's not because they want to go out and be criminals and live immoral lifestyles and do this or that, or, sometimes they've been accused because they wanted to have, more promiscuous lifestyles. It's like, no, I think it's not even that. It's fundamentally that you want to believe that you can figure it out, because the moment you think that something miraculous is at play, it's like, well, you're now at the mercy of something that you'll never understand, that's greater than yourself. And that's not a comfortable feeling, because as a scientist, you want to have some expectation you'll understand and you'll know. You get to the point that you have to say, oh my goodness, this is something that's beyond my comprehension right now and maybe forever. What do I do? Because then you're humbled at that point. And I hear on some of these channels, like, yeah, Sal, you're just finding more things that we're going to figure out eventually. I just want to laugh. I said, yeah. I hear that a lot of times, because that's the response when I come up with something like this. He said, you're just giving us more things we're going to figure out eventually.
Yeah.
It's like in your dreams.
So what is the evolutionary biologist's answer to this topoisomerase problem?
Topoisomerase. So,
topoisomerase.
Okay, now just to give you
I'm really bad at pronunciation, and I don't really care.
No, I'll give you a little bit. This is also educational, because I'm going to share this on my, I'm going to rebroadcast this on my channel. Topo, when they say topological or like topography, it means kind of the geometry or shape or whatever. So you probably hear like topographic maps. So topo, that's why gave the word topo. It's kind of the shape, topo.
Okay.
In chemistry, we can have the same molecule, but then we can reconfigure it different ways. And that's called an isomerization reaction. So when you're seeing that the DNA can be cut apart and untangled and reconnected, that's an isomerization. So topoisomerase. The ending of the word ace means it's an enzyme. That means it creates a certain kind of chemical reaction. So, see, this is what's good about, this is why I enjoy criticizing evolution by just studying basic. This is a very basic science thing of cellular biology. What ends up happening is sometimes the creationists will get sucked into these discussions on the terms of the evolutionists. The evolutionists will be talking about like the tiktaalik, or like what Dessel Drace brought up was the viral stuff. So let's just grant for the sake of argument that common ancestry is real. That's what I often tell creationists. I said, I know you want to argue against common ancestry, but I just recommend just grant that for the sake of argument. It's not that you believe it, but let's just start with that as an assumption. Does finding all this junk in the DNA, does that solve this problem? Does the fact that we're similar in DNA to chimps, and I actually think we are actually more similar than creationists claim. I think we are very, very similar in terms of DNA. It says in Ecclesiastes, men are but beasts. Okay, so that even though we have divine souls, there's a part of us that God reminds us we are mortal. And I'll say, okay, so you could point to all the similarities between humans and chimpanzees, and there are differences that indicate to me that we're miraculously created. But even granting that we're similar to chimps, does this solve this problem of topoisomerase? So, I think I've debated this a long time, and there's no response when I talk about topoisomerase. There's none. Okay, and you could see why. And so I like this much better than talking about bones and similarity. I said, let's look at the function. So this is why the intelligent design movement moved forward. They weren't trying to overturn common ancestry. They were just asking questions like this. How could this evolve? And I'll tell you, when I see this, it's like, doesn't that make you want to worship the creator? Jesus said, consider the lilies of the valley, how they grow, they neither toil nor spin. Yet I tell you, Solomon in all his glory was not arrayed as one of these. And I'll show you why this has more significance now in the 21st century. That statement has more significance now in the 21st century than it probably did at any other time in Christendom. But to do that, I'm going to have to bring up my PowerPoint slide to that effect. And that's going to take maybe a minute on my poor, slow computer.
Okay.
But I will show you. So in the meantime, if you have something to say while this is booting up.
Yeah, well, I just want to thank those of, that disagree with our ideas here, but you're sticking around and you're putting comments in the chat. I really appreciate having people around that stimulate the discussion and that challenge ideas. And I mean, that's really what I'm here to do, is to challenge the ideas that are presented by evolution, even though I am a lay person. But I'll say one thing about, my response, when I asked you what you thought about why do these papers and everything get through peer review without anyone picking up on these critical problems. And even as someone, when I read these scientific papers, I don't understand a lot of what is written, but what I can see happening very often when I read them is that there's always the starting assumption that evolution happened. It's always that there's always embedded in the paper this idea that evolution happened. And so no matter what problems are presented in the paper, no matter what challenges, then that's just like, oh, well, we admit that we can't yet figure this out, but we know that evolution happened. And so there's a lot of, and someone just recently commented to me that evolution is airtight. And I will say this, what is airtight about evolution is the rhetoric in popular media. That is what is airtight. You will not get any doubt when you're listening to popular media or in education. You will never hear them doubt the basic assumption of evolution. It is the rhetoric is airtight. The actual evidence for how all this happened is not at all airtight. And in fact, it is, from what I can tell, every scientific paper that I've been presented with that I have tried to understand, and I'll tell you, as soon as I see a scientific formula, I'm ignoring that. I'm ignoring a lot of what I'm reading. But what I do see is I can tell when it's storytelling and when they're actually putting things that we've observed through experiment. And most of what I'm reading is just storytelling with fancy scientific jargon. So.
Agreed. And if they say it's airtight, what about papers like What's Wrong with Evolutionary Biology, published in 2016 by an evolutionary biologist? What about the third way of evolution that's complaining? And what that paper, What's Wrong with Evolutionary Biology, was pointing out is he was saying that there are all these constant calls for revolutionizing the field. That means that this is not, okay, let's compare this to real theories, real scientific theories, like say electromagnetism, or even something more basic, geometric optics. So when you go to the eye doctor, or they're building glasses, or if they're building telescopes, we know that if you have this certain kind of material and you shape it a certain way, light's gonna be bent a certain way. We don't need, this has been like for 400 years or more. We've never called for a revolution of geometric optics because it is tested science. It is reliable. Everyday practice, we know that it works. We would be shocked if it didn't work. It is so solid. That's what I call an airtight theory. And so going back to Rob Stadler's book about high confidence versus low confidence science, geometric optics would be like at the top of high confidence science. You can put up there also electromagnetic theory. Yeah, there's some things even I question some things because I'm working on the distant starlight problem for young Earth creationism about electromagnetism, but a lot of it's very stable. Your computers are understanding of, like, say, your radio waves, which make your cell phones work, and Wi-Fi, etc. Electromagnetism. You wouldn't have electric generators if we didn't have electromagnetism. You wouldn't have motors. You wouldn't have your telephones. They're just all these devices. It works. That's high quality science. What evolutionary biologists have done is tried to suggest that, evolution science too, so we should accept it as well as electromagnetism, geometric optics, or theories of gravity. And that's not true. Because if you take all of these basic ideas of physics, probability, and then you add the other sciences like, say, cellular biology and like the stuff I was showing you, that's molecular and cell biology, like the topoisomerase, you throw all that in there and you're like, how does that square with evolution? If evolution were a true theory, you ought to be able to take all these other theories and justify it and say, we can independently show that this should work. And it doesn't. And that's why I don't believe in evolutionary biology. Now, I do want to say that there's something you probably noticed this, Rebekah, this whole thing about rhetoric, where they use the term evolution kind of loosely, because, the kids are different than their parents. And you could say, well, the kid evolved, he changed. He's not exactly like his parents. He may have some mutations that his parents didn't have. So yeah, he evolved. And then they'll say, yeah, see, evolution's true, change over time. So if you define evolution as change over time, then it's like even a creationist would be an evolutionist.
And that's an interesting point about the use of genetics, like what we understand about genetics, and calling it evolution. That is evolution of the rhetoric, because 20 years ago, when I started looking into this, nobody was saying all this stuff about, oh, you see, you're different than your parent. That's evolution. No one was saying that at that time. That is kind of like a new development that I've noticed, that, now it's like everything is evolution. Genetics is evolution. So I don't know. Have you noticed that in your time as a...
I don't know. I wouldn't say that I ever heard someone say that you're different than your parents, therefore that's evolution. But what they say is evolution is defined as change over time. Now, there's sometimes they'll say, okay, change in allele frequencies or whatever, but there's so many definitions. So what I'll say is, okay, for the sake of argument, let's just say common ancestry is true. If that's what you mean by evolution, let's say that's true for the sake of argument. Just for the sake of argument, you still have problems requiring miracles. So what are you going to do then? If evolution needs miracles to get the theory to work, then how's this different from creationism? I'll just point out, you guys don't want to admit it, but each time you say this evolved and you imply that it's natural and you can't justify it when it's so obvious that it required a miracle, you really are a creationist. It's what I would call the miracles of evolution that aren't admitted, and I just showed you one example. And remember, I said topoisomerase is only one of the 20,000 some proteins that we presume are expressed. It could be, we don't actually know the number for human beings.
Wow.
But there are all these other amazing proteins. Maybe some are not quite as, kind of have the wow factor of topoisomerase, but I've studied a few and they're pretty amazing. And I'm going to show you some more, but let me try to get my slideshow going, and I could show you even more of the complexity of the topoisomerase. So let me see if I have this. And I'm glad you weighed in, because sometimes it can take a while to get the PowerPoints to get going here.
Well, we're having some little spats in the chat. But
I'm not watching the chat, by the way.
Yeah, I know. Don't. You shouldn't. But I just want to thank Dessel for being here. Spim, Pigs Can Fly. And I think those are the main. Demeter is here, or he was here. I don't know if he still is. But thanks for being here, guys, and sharing your thoughts. And these two military guys or ex-military guys are kind of having it out right now. But so yeah, and I will respond to what Spim said to me. Oh, I see that you're kind of criticizing me for ignoring some of the stuff in the scientific papers. Well, I simply have no idea. I wouldn't be able to understand it. I have to ignore it unless I'm going to go get a degree in math, physics, and all the, I'd have to become an expert in quite a lot of things if I wanted to understand everything in every scientific paper. But as Sal has been doing, when I see that mathematical formula, I don't dispute the math that's being presented there. I accept that whatever they say that formula is showing, I just accept that's true. And so that's not the kind of thing that I'm disputing when I'm looking at these. I'm looking at and trying to understand where the actual evidence is for evolution. And now I'll add Sal's thing.
So there, and I want to, what I wanted to say is there is evidence that would lead some people to believe in evolution. And I've tried to advise people, especially creationists, that don't be too dismissive of them in terms of thinking you can do this with easy arguments or simple. You have to get down a little bit more into the details if you want to have really strong arguments. So I want to thank Mats, if you're listening. He was just showing that picture, the topoisomerase animation to his daughter, his 14-year-old daughter. And I was like, that's great. You see, that didn't take long to get someone thinking. It doesn't matter that you can see the human chimp similarity. And you know, that's another topic why you might think, okay, that's proof of evolution. But I'll say, well, let's just grant that is evidence in favor of evolution. But then what do you do about topoisomerase? Does that solve the topoisomerase problem? You'll say no. So I said, see, so you've got a situation where you have one set of evidence that may lead you to believe evolution, but then you have things like topoisomerase, and I could go on and on about all the other protein things and cellular biology things that show that you need miracles. So it's not quite as airtight as people claim. So now I'm going to give a little bit of a talk on just kind of basic, kind of the molecular level of biology and cell biology, protein biology here, biochemistry, whatever you want to call it. There's this beauty product you could buy at Walgreens called Collagen, and it's supposed to make you look really pretty if you take it. I'm not endorsing this product, but collagen is actually a protein that they're like somewhere on the order of at least 28 types of collagen in the human. It could be more depending on how you classify them. And by weight, maybe about 30% of our protein are collagens. So collagen is a very important protein. So I showed earlier the topoisomerase protein. Here's the collagen. And one reason I want to show it is I want to show you what a protein, if we list it in terms of amino acids, and Rebekah, please feel free to ask questions here. So this is a picture of the spelling of the collagen protein in terms of amino acids. So like, I'm going to mess this up. Like the letter, so this English alphabet here, and each of the alphabetic letters corresponds to an amino acid, even though there are 20 canonical amino acids. We don't fill out the whole alphabet usually. So M would be like, say, you could see like the first letter is an M, and almost all proteins begin with M. There's some exceptions. M is the amino acid methionine. The next letter there is F, which I think is phenylalanine. S is for serine. V is for valine. D is, oh boy, I'm going to embarrass myself. Let me skip that one. There's an R, that's arginine. A is for alanine, et cetera, et cetera. So it does, if I mess that up, someone can correct me. Do you have any questions there?
Oh, I was just going to make sure I'm understanding. So you're saying all of these letters represent an amino acid, which in itself is, you know, a complex chemical, and all of these amino acids lined up together in this order is what constitutes collagen.
Right. So each protein has its own spelling. So the gene provides a way, the DNA provides a blueprint for how to spell the protein. So there's a whole process where it takes the DNA blueprint and then it translates it. It goes through what they call transcription and then translation, and it translates it into the amino acids. So the DNA is the nucleotides, the nucleic acid that is kind of the blueprint. And then there's a process where it takes the blueprint and then implements it in terms of amino acids. So two different, it's a very interesting process. So this is how we make a protein. So this collagen is 1,464 amino acids long, and they have to be in a good sequence. Now there's a pattern here. This is very providential. We happen to have this. I don't know if you could see it, but there's this residue. I may use the term residue or amino acid interchangeably, but there's an amino acid or residue we call glycine represented by G. And there's a pattern here. You can even see it visually. Do you see it now? I highlighted all the G's. So what makes collagen very distinctive and gives it its special properties is that every third amino acid is a G. And there are all sorts of layers of design here. One, that this is an improbable pattern. A random sequence will not make this, okay? It's a violation of the law of large numbers. Some people say, well, that's just like a, some sort of duplication error. I'm just like, no, there are more problems with that. You can invoke that. No need. I can't go into exactly why, but that's also problematic. But suffice to say, you could see kind of like a pattern here already. You can't do this with every protein, okay? Only like a few classes of protein you can do this. But here's an example where you can actually see kind of a hint of the structure. Now, one reason evolutionists will say that there's common descent is you could take the human version of collagen there, and there's a picture of from Casablanca of two humans, Ingrid Bergman, Humphrey Bogart. Their collagen looks like that on the left, and on the right is a zebrafish. The collagen has that same pattern. So evolution will say, well, obviously we look similar, so therefore we evolved. And I say, okay, let's just grant we have common ancestry. That doesn't solve some of the other problems in terms of how you could say maybe, that would explain one thing, but you're left with other problems. So in this case, the collagen between a human and a zebrafish is about 75% identical, depending on the scoring technique. There are various issues with how you can declare something that similar. And we could build these things called phylogenetic trees of similarity. And it's like, okay, that would be compelling evidence in favor of evolution or common ancestry. And I'll grant that. But there are other problems, because then you have other proteins that don't have that pattern. And I've confronted the evolutionists, and I'll say, okay, so you could say that within the same kind of protein, there could be common descent, but between different protein classes, there's not. Because on the right here, we have a zinc finger protein, and you could see it definitely has a different pattern than the collagen. And I say, does that have common ancestor protein? And they'll say, yeah, no, there's no such thing. And I'll say, that's exactly the problem. So right there, you're having a problem with common ancestry at the protein level. That's another story.
Because that would have to be a completely separate line of evolution. And that is, it's already incredibly improbable that it would have come about once. But now you've got to get it, like you have more and more separate lines that have to happen by chance. And that's just...
Right. So this kind of like between, let's say between, okay, a collagen and a human and a collagen and a zebrafish, it's like, okay, I could almost buy the common descent could explain that. Okay, I'll just grant that. What common descent can't explain is you can't have a common ancestor between the collagen here and the zinc finger. You could see the zinc finger is spelled differently. I don't know if you could see the individual letters, but you could see it doesn't have that repeating pattern of the G's that the collagen does. This one has like, I highlight the H's and C's, and the other, the T's, which I think are, I guess that's 3 and 9. All right, so it's a totally different pattern. And I will ask them, do you believe there's a common ancestor, at least in terms of the protein? You might accept common ancestry of the cell, but you can't accept common ancestry of the protein. So just because you evolve one functional protein doesn't mean you're going to evolve all these families from that one ancestor. it has to kind of come from like out of nowhere. I mean, they don't want to say that, but it's like, okay, well, where did this come from? I mean, how did you piece this together? How many simultaneous mutations did you have to go through to make your first zinc finger protein or your first collagen? I said, that's a problem with evolutionary biology right there. And it wasn't in John J. Welch's paper. You should have put it in there. We could make a whole book. I could have given him more reasons to say what's wrong with evolutionary biology. And I'll see that's one of them right there. So there's a problem with common ancestry at the protein level. Now, if you're going to admit problems of common ancestry at the protein level, why can't you start to admit common ancestry problems at every other level? So that's a spelling. And now I'll see if I can go to, I have to correct some of these diagrams because Dr. Dan, the evolutionary biologist, he rightly pointed out some of that's obsolete. So if you ding me for that, that's okay. I'm trying to revise it. So we saw the topoisomerase enzyme in our topoisomerase in that animation. This is how the topoisomerase is spelled. I'm highlighting here kind of the layers of design here. The yellow regions are what we call alpha helices. That's a particular sequence. So these alpha helices have to be put in the right spot. Now point out something kind of cute about this. The topoisomerase, it has to make 2 copies of itself to make a functional topoisomerase. So you see this is the topoisomerase 2 alpha enzyme. And you see that it kind of looks like that scissors that can cut the DNA and do all those incredible things. Well, just like the scissors, do I have a pair of scissors here? You know, each piece is kind of the same. In the topois, you have 2 halves of the scissors. And so you have the 2 halves of the topoisomerase like scissors. It turns out the way this scissor of the topoisomerase is made, you have to make 2 copies of this protein, and then the 2 copies come together to make the scissor, the entire topoisomerase complex. And we call that a dimer, meaning di meaning 2 and mer meaning 2 pieces, 2 polymers or whatever. And they come together. So it has to express itself. So you're talking about foresight of building this. It has to have foresight that not only when it makes this protein, it has to have the foresight that the way it's shaped, that it'll come together to make 2 pieces work as a functioning whole. And I asked biochemists, I said, okay, let's say you didn't have this pattern for a topoisomerase, but I told you had to make a protein that's going to cut the DNA, it's going to untangle it, and then it's going to reconnect it. Can you make a protein just by spelling it that can do all that and do it in only like, say, 1,500 amino acids? Can you solve that problem? Can you take the best chemists, the best scientists in the world, if they didn't know in advance how to build one of these, they can, right now they can cheat because God gave them the blueprint. But if you didn't have the blueprint to make a machine that could do that, could you do it? And I've asked the biochemist who's published in Nature. He said, no way. He said, I don't know of anyone that would even try. I said, yeah. So when Jesus said,
that's what you think happened by chance.
Mm-hmm. Yeah. So when Jesus said, consider the lilies of the valley, how they grow. They neither toil nor spin, yet I tell you, Solomon in all his glory was not arrayed as one of these. Those plant cells need the topoisomerase enzyme just like the humans need it. There's more technology, even just in the topoisomerase enzyme. We're not even talking about the whole plant or the lily of the valley. Then all of our science combined, if you took all the wealth in the world, we could not build a system like this from scratch unless we had the blueprint. Now we can cheat because we have the blueprint by God himself, the great engineer in the sky. So I hope you feel educated now. You've learned a little bit about amino acids and protein biology. You've learned some terms like dimer. This is what they call a homodimer because it's 2 of the same kind. And so you'll hear terms like that, but don't get intimidated. Sometimes just going through one example, you'll be able to read the literature a little bit more. So that's one layer of organization where you see all the colored parts there, the alpha helices, beta strands, and we don't need to go into that right now. But there are other, oh yeah, okay, so my co-author is Joe DeWeese. He's published in the prestigious scientific journal Nature. He's a professed young earth creationist. So how can young earth creationists publish in scientific journals? You see, this is an example. You could be a creationist. You can do biology. You could totally reject evolutionary theory. And by the way, that's another problem for evolutionary biology. How can you be a creationist and do really good biology and you reject what they claim is the central theory of biology? It's well, because it's not really central after all. And it was with Joe DeWeese that I published with, I had the privilege of publishing with him on other papers. And you could see, let me see, there, see Joe DeWeese and me. This diagram was in the FASIB journal in their biochemistry section. This diagram was the one I made. And I'm highlighting kind of the differences across species, and I'm highlighting some of the more complexities of the topoisomerase in humans. Now you see the arrows here, like the colored arrows here, and it's pointing to like the letters. Each of those letters can have like, it's like, can have like a little post-it attached to it. That's like a switch. This is like a programmable protein. So in addition to doing all its marvelous things, it has all these switches that even make it do even more things that we haven't quite figured out. And so I helped publish on this particular project, toward helping cancer research so we could understand the complexity of humans. Humans have a lot more, these little arrows here, what we call post-translational modifications, they have more than any of the other creatures here of those little post-its. That's why that's another example of why humans are special. We are special in God's sight on many levels. And this is just one little tip of the iceberg of it. So not only is topoisomerase marvelous, it's particularly marvelous in human beings. So I was showing that. Now let me show you some more stuff here. This is still the same topoisomerase, but then a different look, different color coding. These are all the functional parts of the topoisomerase. And I don't know if you could see this where it says DQ, DQ, like Dairy Queen, Dairy Queen.
If I make it large, I can. Yeah. Mm-hmm.
Yeah, you will. I apologize that, but just suffice to say there's like this DQ, DQ.
I can see it.
Joe DeWeese is, I mentioned Joe DeWeese, who's my co-author. He's the lead author. His doctoral dissertation was on that DQ sequence because that's where magnesium metals have to attach there. So there are all these critical regions here where like magnesium has to attach to this location. And let me see. The ATPase domain. The green section is where DNA will touch the topoisomerase. So you saw where it's cutting and then splicing and connecting and Okay, those green sections interact with the DNA. That's where it contacts with the DNA.
Wow.
Yeah. Okay, so I say, all right, guys, you think a mindless process can build this? Why don't you try building this from scratch on your own, not working from a template? If you could take the best scientists in your lab and they can't make this, why do you think chance can make this? Why do you think a chance process can make this? And I already pointed out natural selection cannot build this because, the thing will be dead in the process if you don't have all the pieces there. I can't tell you exactly what the probabilities are, because there are some parts you can change and it's not going to kill the topoisomerase, but a lot has to be there. And I'm highlighting this part.
Well, Sal, what they're going to say is, well, you've got millions of years in order to do that. So it didn't happen overnight. So what's your response to that?
What proof do you have that a million years could solve it? We have the problem of combinatorics. Okay, I'll tell you. All right, how many trials do you have to solve it? Because when you have passwords on the order of, like, say, 50 long, and you have to solve it, even if you used all the resources in the universe, you're not going to have enough time. So let's just take the accepted mainstream viewpoint of how old the universe is, how many atoms there are in the visible universe. You won't have the resources. I mean, that is a good exercise. That's a very good question. And I would say that when you actually work through the numbers, it's not sufficient. And this is a known problem in abiogenesis, but it also carries over into what I'd call protein genesis of new protein families like the topoisomerase enzyme. And you can't invoke natural selection for the reasons that we discussed earlier to help solve the problem. You only have chance or something else. And as you pointed out, Rebekah, foresight seems to be foresight and pre-planning and purposeful design seems to be, it just seems written all over this. You know, and I'll point out something. Maybe 50 years ago, maybe 100 years ago, or even as recently as 50, I couldn't have made this argument. I've often said, if I had been, if you transport me where I am now, 100 years back, I might be an atheist because we didn't have the data. But in the 21st century, we have more data now than and evidence of a creator than any time in human history. Even though we live in a very scary time with wars and nuclear weapons, and I think the Lord's return is imminent. We also live in a glorious time, because at the very time we would think that we don't have evidence of God, it's coming to us because we're trying to find cures for our various ailments, medical cures. So God is driving us to discover his creation kind of through the back door. It's because we're trying to find healing. We're discovering our creator. And topoisomerase is a very well-studied enzyme because it's important in finding cures for cancer. So let me see if I could show a little more on this. Do you have any comments, Rebekah? I hope you're getting wowed. Do you have anything to say, Rebekah?
Oh, sorry, I was talking, but I'm really sorry about that. I was muted. I was just saying you're doing an amazing job, Sal. Yeah, I'm just saying wow right now. It's great.
Okay, so now here's an evolutionary problem. Do you see that? You see that red section there?
Yes.
Okay, that's like a password. By the way, this is from a paper I'm working on, and I've gotten some editorial feedback, but the substance of this has been even regarded by, kind of the anti-creationist crowd. They're saying you actually did a pretty good job, Sal. So you see that red section. Just consider that as a password if you can kind of just file that away mentally.
Okay.
Now, the correct term is bipartite nuclear localization signal. Okay, so just think when you hear the word nuclear localization signal, just think password. I'm going to show you a video of nuclear import export, and I'll show you why it's called nuclear. Do you remember? You remember this about the evolution of eukaryotes and prokaryotes?
Yes.
We have the bacteria and archaea are prokaryotes, and even though they may have kind of DNA it may even be in the center like that. In a eukaryote, the nuclear DNA is in what we call the nucleus. And then the nucleus has this membrane that encloses the DNA and other things in it. Any questions? Are you cool with that?
Nope, I'm following. Mm-hmm.
All right. Now I don't have immediately, let me see if I have a example. I don't have a picture of, you can barely see these like little dots here. Those are like little portholes. We call them nuclear pores. So things can go in and out. I'm going to show, okay, so what has to go in and out of the cell here? I mean, of the nucleus? Topoisomerase. Topoisomerase needs to go in and out through here. And I'm going to show the process. And so I have a video I'm going to show of that. So let me load that video. Let me see if I can you, let's see how to, okay, I'll stop sharing this and I'm going to show the video. It's only like 4 minutes long. Now, remember that little red section there I said is the password? Okay.
Yeah.
That's a longer password than what I use for my, when I log into a computer. All right. So let me see if I can, I'm going to get my screen share up and let's see, nuclear import export. Okay. Just give me a moment here. If you want to chat a little bit while I get this all set up. It's going to take me a little while to get the video.
Oh, okay. All right. Well, I don't have much to say right now, but
what are our detractors saying? Have they said anything that refutes my point?
No, they're just, but I mean, they're making a lot of comments that I don't want you to read. And if I had access, I'd delete at least one of them. But so if my husband is watching this right now, please go delete. But so I would ask it to those who are watching, I'm happy with your challenges, but I would ask you to please not attack people personally. It's just, I don't see, if your position is so strong, why would you need to personally attack someone? And I feel like that's kind of a desperation measure when people are faced and confronted with ideas that they don't like. It's just attack, right? So anyway.
All right. So this is what makes eukaryotes special. I mean, this is one of the many things that there are many things that make eukaryotes special, but it's the ability to transport things in and out of the cellular nucleus. So let me play this.
The nuclear pore complexes are the only channels through which small polar molecules, ions, and macromolecules can travel between the nucleus and the cytoplasm. By controlling this traffic, the nuclear pore complex plays a fundamental role in the physiology of all eukaryotic cells. Most proteins and RNAs are too large to move through the complex by passive diffusion and must move by an active process in which appropriate proteins and RNAs are recognized and selectively transported in a specific direction. Let's consider proteins imported from the cytoplasm to the nucleus. Only a subset of proteins can enter the nucleus, and they are allowed in by virtue of having specific amino acid sequences called nuclear localization signals.
I'm sorry, it's a little choppy, but you heard she's using the word nuclear localization signal. And remember in the topoisomerase, that red part, that long password, that's a nuclear localization signal in the topoisomerase.
Okay.
I'll continue now.
Nuclear localization signals typically consist of one or two short segments of basic amino acids. Nuclear localization signals are recognized by nuclear transport receptors called importins, which carry the cargo proteins into the nucleus. With its cargo attached, importin binds to specific nuclear pore proteins in the cytoplasmic filaments. By sequential binding to more interior nuclear pore proteins, the complex is translocated through the nuclear pore. At the nuclear side of the pore, the complex is disrupted by the binding of a protein called RAN to important. RAN carries a molecule of GTP. This binding changes the conformation of important, which then releases its cargo protein into the nucleus. The important RAN complex is then re-exported through the nuclear pore. A protein in the cytoplasm called RAN gap, for GTPase activating protein, stimulates RAN to hydrolyze its GTP to GDP, an action that triggers RAN to release important back into the cytoplasm. RAN plays a key role in protein import and export. Note that while RAN is bound to GDP, it cannot disrupt the binding of important to a cargo protein. In an action that prevents the depletion of RAN from the nucleus, the RAN GDP formed in the cytoplasm is transported back to the nucleus by its own import receptor, a protein called NTF2. In the nucleus, another protein called RAN-GEF, for guanine nucleotide exchange factor, stimulates RAN-GDP to release its GDP and pick up GTP. In this form, RAN-GTP can disrupt the binding of important and its cargo, triggering the release of the cargo in the nucleus. By the actions of RAN-GEF, found only in the nucleus, and RAN-GAP, found only in the cytoplasm, a steep gradient of RAN-GTP and RAN-GDP is maintained across the nuclear membrane, with RAN-GTP inside the nucleus and RAN-GDP in the cytoplasm. Similar to imported proteins, proteins are targeted for export from the nucleus by specific amino acid sequences called nuclear export signals. Nuclear export signals are recognized by receptors within the nucleus called exportins, which direct protein transport through the nuclear pore complex to the cytoplasm. RAN-GTP promotes the formation of stable complexes between exportins and their cargo proteins. Note that this same form of RAN does the opposite for importins and their cargoes. The effect of RAN-GTP binding on exportins dictates the movement of proteins containing nuclear export signals from the nucleus to the cytoplasm. Following transport to the cytosolic side of the nuclear envelope, GTP hydrolysis and release of RAN-GDP leads to dissociation of the cargo protein, which is released into the cytoplasm. Exportins are then recycled through the nuclear pore complex for reuse.
Amazing. Absolutely amazing. You're on mute, Sal.
I wanted to say, as it says in the Psalms, let us praise the Lord for all his wondrous works. And so, they presenting evidence and reasons for the Christian faith, sometimes they call it apologetics. Apologetics is technically defense of the faith, but I view this as building faith. So we can try to defend the building, or we could build the building, so to speak. And I think that we can build the faith of believers and creationists by studying God's work. Jesus said, if you don't believe me, believe the works. And so when I was sharing about cures for doubting Thomas, I said, okay, you look at what God has done in people's lives, the healing, the miracles, the answered prayers, the changed lives. But then we see his works also in creation. And in this, when I studied this, I said, this makes me believe in miracles, because science has shown us that this is improbable. It doesn't assemble it by itself. We know that. We know that there's no reason to expect that. Therefore, something outside of anything that we can explain in terms of science, natural processes. See, we want science to explain everything, and maybe it's not, God did not give us the gift of science to explain everything. It gave us enough to tell us that something beyond science, what science can actually explain, is the cause for our life. And that does, you asked why people accept these papers even though the conclusions are absurd, because the alternative is to accept that something we can't explain is the cause of life. And that's certainly, a scientist lives for being able to explain things. And for a lot of people, that's hard. That's a hard pill to swallow. And, it's like, okay, I came to the point, there's going to be stuff as a matter of principle we cannot explain, but we try to explain whatever we can. But we don't come up with silly explanations like a tornado passing through a junkyard will make a 747. And evolutionary biology and abiogenesis are kind of on that order. You're going to give a silly explanation, and you know that from everything else we know about science that it's not going to work. So at least say you don't know, rather than pretending that you know or that you're going to solve it, because you're not going to solve it. It's becoming evident every day.
Now, you say from everything we know that's not going to happen, right? Like the whole mechanism that you just showed. What is it in science that shows us that is not going to happen by
these are the it's basically the probabilities. And very simply, something as simple as a bolt and a nut. You have to get the shapes just right so that they connect. If you just take random matter and slap it together, it's just not going to connect. So it's just, again, the whole idea, it has to be shaped right to connect right. And we know that the probability of those two pieces is just like a login, a password, a login and a password. And I'm glad I'm mentioning that, because to complete the thought of the topoisomerase, if a bacteria evolved into a eukaryote, those topoisomerases need to have those passwords put in those locations, like that one in red, and it has to be right. There's a nuclear localization signal in the topoisomerase 2 alpha, at least in humans. There's also an export signal. Definitely you need the localization signal. How's that gonna how are you going to reformat the topoisomerase? And by the way, that's only one of the proteins that have to go back inside the cell. There are more things, and I haven't even talking about the mRNAs that have to be exported out there. That's a whole nother process. So you have this gateway. Oh, that's the other thing. You need a gateway. How do you build a gateway that needs like 300 proteins? And then you saw all the other proteins that are shepherding it back and forth and through the channels. Like I can't even name RAN and RAN-GEF, and I just like importins and exportins. Okay, if you don't have that, the cell's dead. And I tried to say, think about that. I wanted to scream when I saw that paper. What's wrong with evolutionary biology? It's like, yeah, you had some good ideas there, John Welch, but why don't you include stuff like that? And I'll say, those are real problems for evolutionary biology.
Yeah.
Just the fact that you could point the fact that chimps and humans look similar, let's just grant that for the sake of argument. That's not going to explain the evolution of a eukaryote.
Mm-hmm.
Not in a billion years.
Yeah.
So that's why it's improbable. I mean, we could go through the exercise of trying to work it out mathematically and rigorously. But from an intuitive level, when you see a cell phone or a car, or even if you've seen some foreign object but its design is because the parts fit. The parts fit together and it works in a kind of coherent way. So even if you don't quite understand what a device is doing, you can still tell it's designed,
Right.
And it took I had to really think about that. I said, how can we actually prove something is, at least improbable? And it's just, it's not like an accident of our perception. Like when you see faces in clouds, it's like, yeah, that's just, we're accidentally just seeing things. But there's some things that are just too well put together. You're like, nah, I don't think that's my imagination, that I think this looks improbable. And I did work through some of the math of that, and just like, yeah, you can demonstrate it mathematically. It's not what we have. Well, our sense of design intuitively is actually kind of not so easy to prove mathematically, but it can be done. And one example is Change Loratan and Rob Stadler's book, Stairway to Life. They actually go through the chemistry and the probabilities, and you're just like, oh my goodness, this is just overwhelming. So
exactly. Yeah, I highly recommend that book to the challengers who are watching. Read this book, The Stairway to Life, and see all the highly improbable, if not impossible, steps that would have to happen for the first life to come about. And I don't know, it's just it's
I hope you've had fun.
I have. Thank you so much. I appreciate your time, Sal. This has been really great. Thanks everybody who's been watching, and we will definitely do it again. So
yeah, I haven't even covered my paper. I think this is more fun than what was in my paper on fitness maximization. That was a really boring topic. But that's that's the thing. It's like there are so many problems with evolutionary biology. It's like, yeah, you have some things that would be, I would put forward even as a creationist and say, that looks kind of, if I didn't know anything else, I'd say that seems like good evidence of common ancestry. But then you have all these other things that tell you, well, okay, you need miracles to make it even feasible.
Yeah.
So,
well, thank you, Sal. Good night, everyone.